Yeah.
Yeah, okay, good.
I can put
Okay.
in the presentation mode. Good.
Okay, great. So, okay, thank you. So, so let me start. So I can thank the organizers for giving me the opportunity, but it will sound strange. But anyway, so it's nice that we have gathered all these people, and I think from different backgrounds. And the idea here is just to give you a short view of the things that we are doing related to carbon and carbon nanotubes. As you know, we can use the carbon nanotubes as sort of uh, filters if they are aligned, right? So this is what we're doing. Let me just go through with, of course, we are talking about carbon. This is American Carbon Society. So regarding that, I would like to invite you to read the Carbon Perspective in 2020 that was just published a few months ago. And our new impact factor of carbon is now 8.82. So it's increasing. It's becoming a very, very nice journal. So you want to contribute, we invite you to submit your papers here. But in a carbon perspective in 2018, we discussed the carbon nanomaterials and the toxicity on this, and this is in particular for carbon nanotubes. So the idea here is you can modify the surface of carbon nanotubes. You can negatively charge, or you can positively charge, depending how you do it. But it has been this correlated to the toxicity of the carbon nanotubes. So negatively charged have represented low cytotoxicity, whereas cationic, so it's higher toxicity. So the point here is, how can we change these charges? How can we make these carbon nanotubes negatively charged? And it happens that we have been working on nitrogen-doped carbon nanotubes, which basically are negatively charged. And then we have done some work in the past. So this is the group members in my group. This picture was taken 1 year ago in spring, so we cannot take it anymore. But these are the people that are working, and I would like to thank them as well. So we have grad students, research associates, and visiting scientists. So the idea here, everything started here with, when I moved to Penn State, is how can we use aligned carbon nanotubes? In this case, is nitrogen-doped carbon nanotubes, and how can we make devices, maybe microfluidic devices, so we can use them for something. And in this case, is for instance for trapping viruses. But before that, we have done some work on the nitrogen-doped carbon nanotubes and the cytotoxicity of these ones. And this is a work that happened in the beginning of the 2000s, and 2007 we published the work. But it was what happens when you put this amoeba, which is a protozoa, unicellular organism, very old and ancient. So the amoeba. What happens if you put different types of tubes, and what happens to this amoeba, right? And the reason was because amoebiasis is a health problem in Mexico, and many people die every year. So what we did, we found that we make dispersions, and this is the control, and then every row here indicates a higher and higher concentration of carbon nanotubes in the suspensions, and then you put the amoeba in the media, right? So we noticed that as time goes by, the cell viability was not really changing in time, and as we increased concentration, the cell viability was still remained quite high. I mean, it was even increased. So this was very surprising, because this only happened with nitrogen-doped carbon nanotubes. And so we did this with other types of nanotubes, 8 different types of carbon nanotubes. And then we noted that this is the usual procedure. So if you try other types of carbon nanotubes, single wall, multi-wall, boron-doped, as you increase the concentration of carbon nanotubes and time, then basically cell viability goes down. So that's a normal situation, but in the case of nitrogen-doped carbon nanotubes was very, very unique. And this is an image of an amoeba that has ingested carbon nanotubes, and you can see the bundles here of carbon nanotubes that are still there. The amoeba is still moving. And we did cross sections of these materials, of this amoeba, and then you can see The nitrogen-doped carbon nanotubes, which show these sort of bamboo-like structures, can be seen here in the bright field image. We have dark field images, and then this is the image. Also, with the other types of tubes, with these multiple tubes that were not doped, we could also see them. So they were ingested by the amoeba. So they were so that sort of was telling us that nitrogen-doped tubes somehow behave better with in terms of toxicity, and there are some other studies with mice that we have done, and some other studies about toxicity with NIOSH, and they seem to be correlated to this. So the nitrogen-doped carbon nanotubes are less, let's say, less toxic. So let's move now to the virus part, and this is what we have been talking all day about it. So basically, what happens when the virus mutates? So you have the envelope protein sort of changes here, and sometimes you change a little bit the RNA or DNA that is inside, but basically it's the envelope protein. So when you have a big mutations, which is called antigenic mutation, your antibodies are no longer effective, and you need to develop new antibodies. So that's when the virus mutates. But the size of the virus does not change in time. So that's a nice thing. So the idea was can we trap the viruses just by size and not by using antibodies, right? So that was the label-free technology. And this is something that we developed. So let me just summarize the work that we have done along in this area. And here we have a substrate where we deposit iron. From the iron, we can grow the carbon nanotubes, and then we can use patterning, and we can just grow the carbon nanotubes here in this tract, so which is 200 microns in width, and the height could be from 30 to 50 to 100 microns length, right? And you have a forest of aligned carbon nanotubes. You have here the substrate, and you can here at the bottom, you can see the metal particles that were responsible for the growth of carbon nanotubes. So this is what we did. So then once you grow the carbon nanotubes in these tracks, you put a polymer, you create a microfluidic device with PDMS, and then you have flow in and flow out. So you can pass through liquids going through, and then they go through this forest of carbon nanotubes. So they go from the inside and it goes outside, and they pass through this wall of carbon nanotubes, which is like a 3-dimensional filter, and the size of this is 1 square centimeter. And this is the microfluidic device. So this is the same, the first experiments we did in 2016, and it took us like 3 years and a half, 4 years to get to that stage. So what is happening here is that you can collect a sample from like a clinical sample from animals or from humans. You pass it through here, this filter, and then you are trapping the viruses. And one thing that we do is we control the intertubular distance of the arrays of tubes, and by controlling that, we can control and we can trap different sizes of viruses. And this is something that we tested, and it's being published. But the whole thing was that at that time we said, we can trap different viruses, herpes or Zika virus or Hep C, influenza as well. So this is some work that we developed at that time. So let me just show you what we're doing. So once you have done this filtering, you can just remove the cap, which is a PDMS, it's a polymer. And then when you look into the SCM, into the scanning electron microscope, this is the H5N2 viruses, and you can see the viruses that are being trapped by the carbon nanotube arrays. But also there's a very strong interaction of the viruses and the carbon nanotubes. So this is a nice thing, and it's quite exciting because we were trapping several viruses and very, very effectively. So what we are doing, so the problem is to have these false negatives. Sometimes patient can be, or animal can be infected, but if it has very low concentrations of viruses, when you do the PCR, you get a false negative. The idea here is that you pass through all the liquid of the sample through the device, and then you concentrate all the viruses in your device, and you can be, and the virus can be detected. And this is the nice thing about this technology. This is just the detection limit and in the PCR detection. And with this device, we were able to increase the detection limit of the PCR by 2 orders of magnitude. So it was 100 times better, even sometimes 600 times much better. So which is very, very exciting. And this is something we already published. So we did, and that was with Yingting Ye, who's currently research professor in the group. We did this with H5N2. And this is, of course, this is influenza flu virus, and this is the way you have the H and N number. And which the diameter of these ones is around 100 nanometers, plus minus. So we have also done work, and this was published with the H11N9, and this again, it was 100% increase in sensitivity. And we did all these clinical tests and also checked the NGS, the next generation sequencing. So we could identify that what we were enriching, what we were trapping, it was this type of viruses. We also did another case study with another turkey virus from the eye, and this is the type of virus that is there. It's a bit narrower, and again, we were able to detect this virus, which was not possible to detect if you just use normal tools like PCR tools. And we got it, in this case, it was just from the eye of the turkey. So you get the tissue, and then you process the tissue, and then you can make a sample, and then, filter it and pass it through the device. Again, this is how we detected. And actually, this was very important because this virus people thought it was a bacteria, and then we discovered it was a virus, and because it was an outbreak here in Ohio. So the nice thing about having these viruses trapped is also, as Yashin was mentioning, it was that these viruses change, and if you do something, they cannot replicate. But the idea here is that once they are trapped in these carbon nanotubes, the viruses are viable. So we're not using an antibody, so it's a label-free system, and the viruses are also viable. So that means you can open your device, you can put your carbon nanotubes with the viruses. You don't have to remove the carbon nanotubes, and you can put it inside a chicken egg, and you can replicate them. And you can also replicate them with cell lines. So this is very interesting because you can show that the replication it's even more, because you have a concentrated sample. So replication it's more effective. So in terms of developing vaccines, this might be a way to move forward. But it's one thing that is interesting is that these viruses are still viable if they are in the device. So this is the way to look at this slide is it's what happens before enrichment and after enrichment. So before enrichment, you have lots of host contamination from the animal, which is not viruses. And in this case, the viral rate, so it was 1.6% of viruses were detected by NGS. But after enrichment with our device, it increased from 1.6% to 90%. So that means this device can enrich very effectively, and most of the sample, it becomes viruses. So this is very, very interesting because you can do other types of tests. And again, the viruses are viable here. So we have a startup on this, and we were working with Nestor on this, and we're trying to make these processing units so we can use them with a pump. So this is with a sort of a pump, mechanical pump, and we can also do it with a manual pump as well for instance, BSL-free facilities. And the idea here is that you pass through very, very, very low volumes per minute, but you can pass through the samples through the device, and enrich samples, and you can detect viruses, right? So we have some collaborations. So let me just mention the next generation of devices that we developed with Team Ye. And this is called Virion. So it's virus capture with rapid Raman spectroscopy detection and identification. So it was a high throughput system. And in this case, this is like 4 centimeters long device. We have the PDMS, you have your inlet and then your outlet, and then you have these herringbone sort of arrays of carbon nanotubes that we're using. In this case, since we're using Raman spectroscopy, we also deposit gold nanoparticles on the carbon nanotubes to get an enhancement in the Raman signal. But the idea is just to trap viruses in a similar way, enrich a sample, and try to see if we can differentiate viruses using Raman spectroscopy. And this is something that we published recently in PNAS. So the device is a little bit different because you have here a gap. So you don't have closed this, so you have a gap. So your fluid goes here. There's some turbulent fluid going there. So you have your viruses, your gold nanoparticles, and then your Raman signal. And the idea is said, well, can you identify using, I mean, these viruses using Raman spectroscopy? And this is something that we have been doing. So in this device, the nice thing about this device as well is you can have different zones with different intertubular distances. So you could trap large viruses, medium-sized viruses, and small-sized viruses, right? And at the same time, this device, it has many functions. So you can open your device, you have trapped the viruses, and then you can do the cell propagation. You can infect cells here, and these are the viruses and the cells. And you can propagate and replicate the viruses just from the chip, right? So this is a nice thing about this. In this case, this is the human parainfluenza virus 3, which we tested here. So again, this is a device that has different, is very versatile. Because let me show you. So you first make the device, you trap the viruses here. You can do label-free Raman spectroscopy, which you can do it with the device, cap device with the PDMS. And after you did the Raman spectroscopy, and then here you have the Raman signals of different types of viruses and subtypes of viruses. And then we use machine learning to classify the different types of viruses. But after the Raman spectroscopy, you can get it's positive. Then you have the sample with the viruses enriched that it's also these viruses are viable. So you can do, after you have these viruses, you can do virus propagation, replication, or you can do genomic sequencing or immunostaining, and you can have other molecular biology tests on the same device, the same cartridge. So this is, let me just show you what we have done. So with the human respiratory viruses, we tried the rhinovirus, the influenza, the parainfluenza, and this is the Raman spectrum for these are different. And using machine learning algorithm, we can really classify and identify different viruses. And the accuracy is around 92%, depending on the viruses and how good the Raman signal is. So right now we're trying to improve the signal to noise ratio of the Raman signal and trying to use different platforms for doing this. But it's in a matter of 2 minutes, you can really screen a sample and you can say, well, it looks like it has this type of virus. Of course, it has to be on your database. But then if that works, then it's very, very, very helpful. So this is something that we have done recently with the chicken coronavirus. So it's the infectious bronchitis virus. And in this case, these are the SEM images. So you have the carbon nanotubes and you have the virus that is being trapped. This is a coronavirus. So we wanted to see if the coronaviruses were different from the flu viruses. Of course, the structures are different and you have this spike protein. So we managed to get this working, and this is just to show you that this is just preliminary data from the chicken coronavirus, but here you can see the peaks coming from the chicken coronavirus. These are two different laser excitations. These broad peaks are coming from the carbon nanotubes, but you can see these peaks, which are very different from all these peaks that were in the other ones. So actually the accuracy for identifying these coronaviruses is much higher because we have more defined peaks compared to the others. So I think this is something we're still working on it. And of course, we're now trying to test and eventually trying to do Raman spectroscopy of the COVID-19, because I think it will be very, very important. So we have an NSF grant on Convergence, and this is the vision that we have for the whole thing, right? So we can really have a fast testing, and you have a 5G cloud where you can process these cartridges and what is coming from this cartridge with your Raman spectrometer. You can really monitor, let's say, hospitals, airports, people, and you can maybe build a database at the worldwide level. I mean, that's a very, vision looking forward, but this hopefully will be something that we can work on. And of course, there are different types of sensors, but I think this technology, it's something that it's very versatile, and we believe it will be developed in the future. So we also, we have a Penn State seed project on this. We're trying to improve the signal to noise ratio of this, and we're using now a 2D metal, which has other types of properties, very new properties. So you can have 2D metals with silver. So it's 1 atom thick layer of silver, which is capped with the epitaxial graphene. And when we did this with the H5N2, then we could see the signals were much sharper and more distinct compared to the normal device that I presented. Again, the changes in the spike proteins, the surface proteins, we believe we're getting more information from the groups from the proteins, also from the lipid bilayer, we're getting information. We have some groups there, and we're trying now to understand more the Raman spectrum. But what we know using the machine learning is that we can classify different types of viruses because the Raman signals are very similar. And this is a new project as well that we are trying to make things more smaller. Can we make a Raman spectrometer which is, like, let's say, handheld? And together with Sona Hosseini, and she's at the Jet Propulsion Lab, so she's working at NASA, and she's making these spectrometers for the space exploration, right? They want to detect, using this spectrometer, want to detect water on Mars, or want to detect water in the moon, right? And this is what we are working on, right? So this is a quarter, and you can see it's very, very light. So I think this is moving forward, and this is what we would like to do in the future. Of course, this will not address the current problem with the current pandemic, but I think this technology might be used in the future for monitoring different types of, monitoring the environment or people, right? So just before I finish, because I still have some time, and I want to give some time for questions and also try to save some time as well. So, and this is just addressing what Yashing Huang was mentioning, development of these self-sanitizing surfaces, antiviral sprays, antiviral surfaces, virus sensors as well, filters for masks. Can we use these 2D materials? Can we use this functionalization of metals like copper or silver, and can we make these systems or these materials antiviral? Can we develop these antiviral surfaces? So we're starting to work this from, this is one of the centers we have, Atomic Center for Atomic Within Multifunctional Coatings. And we're trying to move into these directions. And if you are interested, we will be happy also to discuss in detail. But here, this is some of the recent work we have. So we have single metal atoms, in this case, gold or nickel, on top of the moly disulfide, and they are pretty stable. And there's sort of a weak bonding with the sulfur and this gold. And we're just working on this because we don't know what are the antiviral properties of these systems, if there are any, right? Because as you reduce the size of the particles, the antiviral properties could be even enhanced further. So we're trying to understand this, but again, this is how maybe material science can help addressing some issues related to the current pandemic and maybe also to future pandemics. So I think I would like to thank you and to thank our sponsors and all these people that we're working on different projects on this, but just wanted to give you a very quick overview. I'm trying to save time. So I think it's 25 minutes. And you have questions, you can send me an email or you can do some chat. So with that, I finish my presentation and I thank you for your attention and for being here.
Thank you, Mauricio. I think there are two questions. One is, are these devices reusable? I think talking about the carbon nanotube device that can trap the virus.
They are one-time use, but I think in terms of using it, I think it's you can basically you get sample from a patient, and that sample from that patient is still in the device, and then you can do other types of analysis from the same device. So it's a disposable cartridge. That's the concept we have.
Okay. There's another question. How does the bonding occur? This is a chemical bonding or physical bonding?
That's a very good question. We believe there's a strong interaction of some of the surface proteins with these nitrogen-doped carbon nanotubes, because there's some also deformation. So we're still, there's some sort of strong interactions of the surface of the virus with the carbon nanotubes. We're trying to understand more. We're trying to do high-resolution electron microscopy, trying to see what happens there. But the fact is that if the viruses are viable, that's still, we're not changing much of the structure as well, right? Sometimes if you change things, then if you fold a protein or something, maybe that's, your virus is inactive. So that's further work needs to be done along these directions. And we can also try to anchor, for instance, antibodies to the carbon nanotubes. And if we do that, we can be specific on the types of viruses that you can detect. We haven't done that so far. But yeah, there are many things we need to have more clarifications. But there's something happening with the nitrogen-doped tubes, and we haven't done it with the undoped tubes, but I think that's also something we should try in the future.
Okay, thank you, Mauricio.